My name's Olivia, and today I'll be showing you how to dissect the central brain of a fruit fly with the ventral nerve cord attached. Because the brains are so tiny, you'll need a dissecting scope to see what you're doing. I like to start a dissection with my materials close by. I use a few different tools and reagents in this protocol, including forceps, a Siligard dissection dish, a transfer pipette, and PBS, as well as some ethanol, which I'll get to in a second. We line Flora dishes with Siligard in the lab to make our dissection dishes, which protects our forceps from damage. The cap of this dish is a convenient container for ethanol, which is how we will euthanize the flies before dissection. Ethanol also breaks down the waxy cuticle of the fly, so that when we transfer the flies from the ethanol into the PBS, they're easier to dissect. The entirety of the brain dissection occurs in PBS, which is essentially a salty solution that keeps the tissues hydrated and prevents desiccation. I transfer the slightest amount of PBS to create a dome-like structure over my Siligard dish before placing it underneath my scope. Next comes the flies. I find it easier and a little bit cleaner to transfer a small portion of the flies into a smaller vial. Once the flies are transferred to a smaller vial and the lid's on tight, I then move this vial to ice. The ice puts the flies to sleep, so you can handle them one at a time without all the others flying away. Holding the vial of knocked out flies above my container of ethanol, I gently tap on the vial until one fly is transferred to the ethanol. You'll want to leave the fly submerged in ethanol for around 30 seconds to ensure that the fly is euthanized and to break down the waxy coat of the cuticle. After no longer than 45 seconds, you can transfer your fly from your ethanol dish into the PBS on your Siligard dissecting dish. I recommend playing around with the magnification until you find a view that suits you best. To start the dissection, you'll first want to remove all of the appendages coming from the thorax, including the wings and all 6 legs. You'll be dissecting away most of the fly's body during this process, so I find it most helpful to keep my dissection dish as clean as possible by removing most of the body parts from the dissection dish. In this video, I've cut out all the moments of time where I'm wiping my forceps off on a wet chem wipe that is outside of the field of view of the dissection scope. I recommend doing this yourself, as it keeps your forceps clean and your dissection smoother. Once all the appendages are removed from the thorax, you'll want to dissect the abdomen away. I find this easiest by grabbing on to the scutellum, and with a firm grip on the abdomen, I pull the two apart. Most of the time I spend dissecting is focused on trying to get the best grip with my forceps. Here, I am trying to grip the scutellum with both of my forceps, but in the process, I remove a couple bits of cuticle from the thorax. Once I grip the scutellum with both of my forceps, I pull my forceps apart to split the thorax up the middle. I find splitting the cuticle up the thorax makes removing smaller pieces of the cuticle easier. As you dissect away pieces of the cuticle from the thorax, you'll be able to see flight muscles and other internal organs. The flight muscles are slightly more opaque than the neural tissues. They are also dorsal to the ventral nerve cord, so it's pretty easy to peel these away without damaging the ventral nerve cord. The trachea, which is the reflective white that you've seen, is how the insect is able to do gas exchange within their body. You can safely remove the trachea without damaging the neural tissues. You'll want to make sure all the last bits of cuticle are removed from the thorax. As you remove more and more of the cuticle, the ventral nerve cord will start to come into view, and then you can start to clean up all the other debris surrounding it. It takes quite a bit of practice to start recognizing these structures, so be easy on yourself. I find the cuticle surrounding the neck connective tissue to be some of the hardest to remove without damaging the ventral nerve cord. So I try to be extremely cautious when removing cuticle anywhere close to this region. Just a few more final touches and the ventral nerve cord will be fully dissected. You can see me here trying to grab for the proboscis, which is just to try and get me a better angle to dissect the rest of the debris away from the ventral nerve cord. Now that the ventral nerve cord is dissected, I move to the head capsule to dissect the central brain. The first step in a central brain dissection is to remove the proboscis. Then I like to try to grab for the eye with both forceps to try and split it up the middle. But sometimes other pieces of cuticle get torn off in this process, which is totally fine too. Splitting the cuticle, whether it's on the eye or elsewhere, just makes it easier to remove bigger pieces of cuticle from the head capsule. I got pretty lucky with a big piece of cuticle coming off with one tear, and so now I'm just working on getting the rest of the cuticle, which seems to be a lot of the eye, removed from the central brain tissue. At this point, you should be able to tell the difference between neural tissues and cuticle, which makes it a little bit easier to grab even when your brain seems a little bit out of focus. Now that all the cuticle has been stripped away, you can now see the differences between the neural tissue and the trachea surrounding the brain, which is that white reflective matter that I'm stripping away from the central brain. Sometimes the trachea can have a pretty good grip on the brain, and so I like to use trachea on opposing sides of the brain to try and separate the two. Once all the cuticle is stripped away, I find the brain to become quite sticky. So you can enjoy the last few seconds of this video watching me struggle to get my forceps unstuck from the brain without inflicting any damage. This can be a little tricky when you don't have much of anything else to hold on to with your other forceps. Finally, a full brain dissection with the ventral nerve cord attached. Thanks for watching. I hope this was helpful. If you have any questions, feel free to leave them in the comments below. Oh, and follow me on Instagram @LivLovesBugs.